I'd like to welcome you to the first How to Talk, formerly known as Postdoc Talks. This is our third series. We're starting off this one with Dr. Tatiana Garcia-Bates, who is from the Robert Ferris lab, and she is currently working on adenoviral vector dendritic cell vaccines to target tumor cells. And today she'll be talking about ELISPOT assays. So, Dr. Garcia-Bates, please.
Thank you so much for coming to this talk. So I'm going to be talking about how to do an ELISPOT assay to detect immune responses in vitro. But I'm going to be focusing mainly on the humoral immune responses to looking at ELISPOT assays, just because that's kind of what my expertise is more about. Before I go into how the assay is done, I would like to give a brief introduction into how immune responses happen in the body after they encounter a microorganism. So there are two types of immune responses: the innate immune response, which happens usually immediately within the first 4 days after encountering an antigen. And this type of immunity is very important to defend our body against invading microorganisms. However, this response is mainly non-specific, and it requires for the microorganism to have special recognition patterns on their surface to be able to be recognized by these innate immune cells. So, even though this innate immune response is very important, the immune system needed to evolve to be able to recognize a wide variety of antigens that come from viruses, bacteria, and so that's when the adaptive immune response came into place. Adaptive immune responses usually happen after 4 days of encountering an antigen. And usually happens because the antigens are transported into the lymphoid organs, and there they are recognized by these adaptive immune response cells that are called B cells and T cells. They there undergo clonal expansion and differentiation into effector cells that are there going to be able to remove the infectious agent. And these adaptive immune responses, as I said before, they are more antigen specific, so therefore are better at removing the infectious agent. So there are two arms of the adaptive immune response. And first I want to point out that these cells not only can do adaptive immune responses, but they can also do innate immune responses. But I'm going to focus it on the adaptive immune response of these two arms of the immune response. So one arm is what we call humoral immunity. And the humoral immunity is basically done by B lymphocytes that are able to recognize antigen through the B cell receptor in their surface. Once they recognize the antigen, they undergo clonal expansion and they differentiate into the effector cells called antibody-secreting cells. These antibody-secreting cells can be also called plasmablasts, or they can also be called plasma cells or immunoglobulin-secreting cells. So, for the other arm of the adaptive immune response, we call cell-mediated immunity. And this cell-mediated immunity is mainly done by T lymphocytes. And the reason these evolved to have cell-mediated immunity is because some antigens are intracellular, so they needed to be recognized. And therefore, to be able to recognize these intracellular pathogens, the T cells are able to recognize the peptides made by these antigen-presenting cells and are able to be expanded and produce cytokines that then will differentiate these cells into effector T cells that will then target the infected cell and do target cell killing. So for the purpose of my talk, I wanted to look at how we can detect antibody-secreting cells in peripheral blood from individual that is undergoing a current infection or even after vaccination. And I also will touch a little bit about how we can detect cytokine secretion from T cells, and again in the same setting of vaccine or immune responses. So first, I would like to focus to you again, just looking at the humoral immune response. Again, how we can detect antibody secreting cells. Here is just an example of how an antibody response occurs during an infection or immunization. In the top panel, I'm showing you the concentration of antibodies over time. And here I'm showing you, this is the time after immunization in weeks. So after the first immunization, you can see that the first antibody that comes up is the IgM. And this is the antibody isotype that is mainly produced in the primary immune responses. However, these antibodies wane if you don't have a second boosting immunization or a third boosting immunization. So that's why you do these 2 booster immunizations to increase the amount of antibodies in the serum. And mainly, the most important one will be the IgG, which not only is important for protection, but also has a higher antibody affinity that would recognize the antigen better than any other isotypes. So the antibody affinity is important because as the memory B cells get reactivated, they undergo class switch recombination and somatic hypermutation to develop these B cell receptors that are have a high affinity for the antigen that they are being exposed to. So, yes, this is the way that currently, vaccines are being tested. Just looking at antibodies in serum of individuals that receive a vaccine. However, there are other ways that we can look at antibody responses right after an immunization, and the better way to do this is by using an ELISPOT for direct ex vivo humoral immune responses in blood. So this was a paper that just recently came out in Nature, where they developed an ELISPOT assay to use an ex vivo using whole blood or PBMCs isolated from people that have been vaccinated. And so they are able to see that most of the antibody secreting cells come up at a window of 4 to 7 days after a secondary immunization, as you can see here. And the importance of this is that this happens earlier than what you see in the serology or antibodies in the serum, that you always have to wait about 4 to 6 weeks after immunization or after boosting. So, and the other important thing is that when you use antibody secreting cells, detection of antibody secreting cells, you get rid of the background antibodies that you have, the baseline serological baseline that you have of previous exposures to antigen. So therefore, this is a more sensitive assay to look at vaccine efficacy of, against a new vaccine. So this is just an example of how antibody secreting cells in circulation are found during an acute dengue infection. And this is part of the research that I did in my first postdoctoral experience at the Center for Vaccine Research. I worked with a mosquito-borne viral disease called dengue fever, and we had patient samples, or PBMC patient samples, that were flown to us here in Pittsburgh from Brazil, from a well-characterized cohort of patients. And I was able to look in these patients during the acute phase of the disease, which is usually between 4 to 7 days of exposure or developing symptoms. And what I found is that, again, on healthy individuals and individuals that have fever of unknown origin or different origin than dengue, you don't see that many plasmablasts, which are shown here in red. They have lower levels of CD20 and higher levels of CD38, and you can see them by flow cytometry. So what we found interesting is that in patients that have dengue fever or complicated dengue fever, which is very similar to dengue hemorrhagic fever, you see a peak of antibody secreting cells, and this peak usually happens between day 4 to 7, and especially on secondary immune responses. So then again, this assay could be possibly used to as a diagnosis assay to detect a recent or current acute dengue infection. And not only we can do it by flow cytometry, but we can also do it by ELISPOT assay. And I will show you that data later. But okay, now that we're talking about ELISPOT, what ELISPOT stands for? So ELISPOT is an enzyme-linked immunospot and was originally developed by Cecile Serkinski in Sweden in 1983. It was originally developed to enumerate antigen-specific antibody-secreting cells. And it subsequently was modified to also detect cytokine secreting cells. So, what are the advantages of an ELISPOT versus the ELISA? As we know, ELISA is being used for years to detect antibodies in the serum or in the soups of PBMCs incubated in culture. But one of the advantages of the ELISPOT compared to ELISA is that the results are given quantitatively, because you can see the number, or you can say the results as the number of cells secreting antibody or cytokine of interest, while the ELISA is more of a concentration of antibody cytokine produced by all of the cells in culture, and is usually given by picograms or nanograms. Here is more cells per million, interferon gamma or IgG secreting cells per million cells. So it's a more quantitative assay. Also, with the ELISPOT, you're able to detect 2 proteins secreted by the stem cell, while with an ELISA, you cannot do that. The detection of color for an ELISPOT is usually done in the same culture plate where they are being cultured. And in an ELISA, you have to collect the supernatant of your culture and then do the ELISA on a separate plate. The analysis of the ELISPOT is usually done in a dry phase, while the ELISA is done in a liquid phase. The storage time and possibility of reanalysis. So the ELISPOT, you can actually reanalyze those ELISPOT plates up to like months after you develop the assay, while the ELISA, you pretty much have to like read it right away. And the bias due to consumption of the protein by culture cells, and this is mainly on when you're looking at cytokines, you don't have the bias on an ELISPOT because the cytokine is captured by the ELISPOT, while the ELISA, some of these cytokines are being used by the cells themselves. And this is why the sensitivity of the ELISPOT it has high sensitivity, while the ELISA has a low sensitivity compared to the ELISPOT. So here is just showing you the experimental design of how the ELISPOT is done. So the assay consists of a series of steps. The first one is the preparation of the solid phase or the coating. The second one will be the isolation and the counting of the cells. The third step will be the incubation of the cells. The fourth is the immunoenzyme detection of the spots. And fifth, last, is the spot enumeration by an ELISPOT plate reader. So this is just an schematic diagram of a one-step or two-step antibody secreting cell ELISPOT. And again, this is taken from the Nature Protocols paper in 2013. They developed a way to look at vaccine volunteers where they can actually take their whole blood and either lyse the red blood cells and use the cells in the whole blood, or they isolate PBMCs from using the FICO gradient centrifugation. They also are able to enrich for antibody secreting cells using magnetic beads that are against antigens in the cell surface that are only found on B cells or on antibody secreting cells. And on the one step, they put together the cells with the secondary antibodies, But one drawback of this one is that the background is higher and you have to do the assay for shorter periods of time, only 3 hours. While the two-step, you can do longer periods of incubation up to 24 hours to look at cytokine secretion or, in this case, antibody secretion. The nice thing is that you can use 2 different substrates to be able to detect one type of immunoglobulin compared to another type of immunoglobulin. So you can see some of the blue ones would be, for example, IgN, and some of the red ones would be IgG. So that's another advantage of doing an ELISPOT. But just to go over all of these steps very carefully, first step is the coating of the plate or the solid phase. The ELISPOT plates that we use are either are clear polystyrene wells or wells whose bottom surface are made of high binding capacity membrane, and is usually the PVDF membrane or nitrocellulose membrane. The binding of the proteins to this membrane are usually hydrophobic interactions, so they are very difficult to take out of the membrane. The plates can be coated with either an antibody against the isotype antibody of interest, for example, anti-human IgG or an anti-human IgA, or they can be coated with the antigen itself, for example, the virus particle or peptides. And this is mainly in the case when you're looking at interferon gamma responses from T cells. But you can also look at antibody responses against viral particles. The coating is generally done overnight at 4 degrees, but you can also alternatively coat the plate for 2 hours at 37, or you can coat it at room temperature for 4 hours. The paper that I just showed you now also developed a way of coating the plate and blocking it and then drying it out, and you can store those at room temperature for long periods of time, up to 6 months. So you could potentially use this as a high throughput of this assay when you're looking at vaccinated people. So before adding the cells, the plates need to be incubated with a blocking buffer, and this is obviously to prevent non-specific binding of secreted antibodies. And the blocking can be done with either BSA, fetal bovine serum, or skim milk. And you just have to try different blocking reagents to see which one works better for your specific assay. For me, when I was looking at antibody responses, using skim milk was better. But when I was looking at interferon gamma responses, using 10% human serum was better to do.
So is that just empirically determined?
Yeah, that's basically, you just have to try the different blocking reagents depending on what are you looking at. And these individuals here tested different ways of doing this ELISPOT, and one of the things that is important to know is that the coating concentration of the antibody that you use to coat the plate is very important. So you probably have to do a series of dilutions to determine what dilution is better for your coating antibody. I had to do that when I was doing PBMC or B-cell ELISPOTs, looking at dengue specific responses. I needed to titrate how much, in my case, the viral virion, how much I needed to put to actually get the same spots across the board. But again, so if you use 1-to-1 dilution, you get a better spot definition, while if you dilute it too much, then you might not see a good spot definition. Also, it is important to see the influences of blocking of reagents. This one was blocked with BSA, while this one was blocked with 2% skim milk. So again, I have found that skim milk is good for looking at antibody responses, but then for interferon gamma, I found that human serum was better. So I just play with what's better to give me a better result, or a more clear result. It is also important to use affinity purified antibodies, and it's better to use them against the light change of the antibody, either the kappa or lambda chain of the antibody. And this is superior than using a whole IgG, antibodies against the whole IgG fraction. So now the next step is the cell isolation and the preparation of the cells. So the assay can be performed with PBMCs. They could be either isolated by the gradient centrifugation or using phycopaque, or in other cases you can use whole blood. And the reason these researchers develop a way to use whole blood is because sometimes when you have, especially pediatric patients where you don't have that much blood to work with, using whole blood will, decrease that amount of cells that you need for your assay. So when you use whole blood, you obviously need to lyse the red blood cells first before using the rest of the cells for your assay. One important thing is these assays can also be performed with either freshly isolated PBMCs, or they can also be used with frozen PBMCs. They have done comparisons and both of them work fine, especially for antibody-secreting assays. The B-cell ELISPOT can be done to detect circulating antibody-secreting cells at the peak of infection and immunization, meaning between 4 to 7 days after a secondary infection. But they can also be done to detect mitogen reactivated memory B cells. So you can actually look at memory B cell responses by reactivating them in vitro for 4 to 7 days, and then taking those cells and putting them on an ELISPOT for 18 hours to detect what antibodies and what are they specific for. So again, this is looking at memory B cell responses, while this is looking at the actual response that is going on during the acute phase. So here's again showing you just a summary of what they did, where they actually are able to use lyse whole blood or PBMCs, and they use magnetic coated beads. In this case, they use CD19 and HLA-DR, which are highly expressed on B cells. And as you can see here, the positive fraction is giving most of the IgG secreting cells, while the negative fraction is basically doesn't have any spots. So by doing the enrichment, you will help increase the sensitivity of the assay, because obviously you don't need to use as many cells for well. But when you do positive cell, I have never done this for my assays. And mainly is because obviously the samples that I was getting from Brazil didn't have too many cells to begin with. So if you do magnetic isolation, you might decrease the yield of the cells that you start with. So you just have, obviously because these ones are freshly isolated PBMCs from donors, it's easier to do this magnetic enrichment technique. The cell incubation stage in the development. So depending on whether you're doing a one-step ELISPOT or a two-step ELISPOT, the incubation time may vary. So when I show you I'm just going to go back for a second. When I show you the one-step ELISPOT assay, they add the cells together with the secondary antibody that is conjugated to HRP, for example, horseradish peroxidase or alkaline phosphatase, and they incubate both of them together. So there is a little bit of higher background when you do this because you don't have the additional washing steps. So for this type of assay, you probably can only do it for 3 hours to detect whatever they are being secreted at the time frame of 3 hours. But if you're doing a two-step where you add the cells first and then you wash the cells off and then come back with a secondary antibody conjugated with HRP or AP, then you can actually do this for a little longer periods of time, like 18 hours or 24 hours, to detect both either cytokine secretion or antibody secretion in this case. So again, this I already said, the two different, also it's important to use 2 different cell densities. I at least do 3 different cell densities because you don't want to have too many spots that you cannot count them, and you don't want to have too little that there is nothing to count. So depending on whether you're using PBMCs or whether you're using magnetic enriched populations, you will use different 10,000, 50,000, and 100,000 cells, and just to see which one will give you the best spot definition. Last one is the immunoassay development. So after the cell incubations, cells must be carefully removed and washed with detergent-free buffer first, and then follow it by detergent-containing buffer. And this step is important. I mean, all of the washes need to be done with a little bit of detergent. You don't want too much that the membrane will become permeable, and then a lot of the wells start leaking. So usually between 0.05 or 0.1 twin 20 is enough to get rid of the cell debris and get rid of the background, but not compromise the membrane permeability. So again, and it's at least 5 washes in between the steps. Then you come in with an isotype-specific secondary antibody in the case of the 2-step ELISPOT. And you could use the HRP conjugated antibody or the alkaline phosphatase conjugated antibody. And when you use these two together, you can actually look at different cytokines at the same time, or at different antibody isotopes at the same time, because you can have a secondary against IgM, for example, in alkaline phosphatase, and an IgG, for example, against HRP, and then use different substrate, and then you get either a red or a black spot, depending on which isotype you're looking at. So the substrates are mainly commonly used for HRP are the AEC and the TMB in conjunction with a hydrogen peroxide. Sorry. Yes. And this is just give you an example of what troubleshooting or some of the issues that you could run into when doing an ELISPOT. So this is how a usual ELISPOT should look like when you're doing a dual color one. Some of these ones that are shown here in the magnified area here are artifacts. So you shouldn't count those that look like fuzzy and not really like a spot. This is just a negative control. And these two here happen when the cells are rolling. There is like a rolling effect. And so one important thing when you're doing an ELISPOT, you gotta make sure that you have a vibration-free incubator. And also, like, I usually put a sign on the incubator, please don't slam the door, so that, like the cells don't move when you're slamming the door of the incubator. Sometimes people just close the door too hard, and then that can lead to this type of movements. Because the cells need to be kind of in the same spot to make the cytokines or the antibodies. So if they move them, then these things can happen. This is obviously too much, too little wash, and that's why sometimes you see a lot of cell debris. And this is too much development. So if you also leave the substrate for too long, it might lead to higher background, because so you also have to watch when you add the substrate. Always watch the spot formation, and if it just becomes too blue, you just have to wash it with water right away before it becomes too blue that you cannot distinguish the spots. So those are, and this is just a little bit of showing you a little bit of what I did with ELISPOT on my research in the Center for Vaccine Research, where I work with dengue, and again showing you some of the stuff that I did over there. So again, I talked to you about how we got frozen patient PBMCs from individuals from Brazil that were undergoing an active dengue infection. And then I thought these PBMCs count the cells, and I coded the ELISPOT. And this is just looking at the antibody secreting cells without prior simulation. So I either was looking at total IgG secreting cells, or I wanted to see whether they're specific for dengue. So I used 3 different dengue serotypes, dengue 1, dengue 2, and dengue 3, and see which ones are recognizing. And when I'm looking at total IgG secreting cells, I code with an anti-human IgG. When I'm looking at dengue specific cells, antibody secreting cells, I use dengue virion to code the plate. And again, I had to do a titration of how much virion I had to put on the bottom of the well to have the best spot formation. I then added the stimulated cells in vivo, and then I let it go for 18 hours where the antibodies will bind, and then I will again detect by using a bioattenuated antibody and streptavidin conjugated to alkaline phosphatase to develop the spots. So this is kind of what data that I got. It was published in January of 2013. I found that a lot of these patients that have secondary dengue hemorrhagic fever type of disease during the acute phase had high levels of IgG secreting cells. This is only having 1,000 PBNCs on this well. And most of them were dengue 3 specific. There was not that much of a bystander activation of the B cells because we didn't see any IgG recognizing influenza or BSA, so these ones are irrelevant antigens. And in a healthy individual, you can see that there is not that antibody secreting cells on a healthy individual. Again, these patients are also from Brazil that are healthy. And this is just comparing, having all of the spots. There are some influenza-specific antibody secreting cells, but mainly all of them are against dengue 3, specific against dengue 3. I also did another antibody secreting cell assay using an ELISPOT where I had dengue 3, dengue 1, and dengue 2 just to compare what they were infected with. And you can actually see what serotype they're being infected by looking at the response against the different serotypes. And so just to let you know, the ELISPOT has also been adapted to other immune monitoring, and I'm not going to go into how to do these T-cell assays, but just to let you know, the ELISPOT can also be used for interferon gamma secretion by T-cells, especially CD8 T-cells, but it can detect both CD4 and CD8s in response to an antigenic or hematogenic stimuli. Interferon gamma is produced in high quantities mainly by Th1 cells, and is frequently used to track CD8 T-cell responses, as I said before. While interferon gamma secreting antigen-specific T-cells can be detected directly from peripheral blood, sometimes these T-cells need to be requiring in vitro expansion, or what we call in vitro stimulation, to augment the low-level responses, especially for the specific T cells that are in very low amounts in the peripheral blood. So you might have to do a two-step incubation with dendritic cells loaded with peptides of whatever specificity you want the T cells to be, and then test for interferon gamma responses against these peptides using the ELISPOT. Another adaptation was to use this ELISPOT to look at gran enzyme B to detect cell cytotoxicity by CD8 T cells. And this is also important because CD8 T cells not only can secrete interferon gamma, but also they secrete gran enzyme B when they see the antigen, either by a dendritic cell or presented by a tumor cell, for example. So this is another way to look at functionality of these CD8 T cells against a specific antigen. And so this again, I just wanted to show you like the differences between the antibody versus the cytokine is that we're looking at the cytokine released by the cells that are stimulated either by a dendritic cell or they are stimulated by CD3 or CD28 beads. And again, you use the same streptavidin enzyme conjugate, and you can see a spot generated for every single cell that is producing interferon gamma. So what are the improvements made over the last 2 decades on ELISPOT assays? As you can see, they are now using hydrophobic membranes. They didn't used to have these hydrophobic membranes before. And they obviously passed it to a miniature 96 well plate format, so now you can do a lot of wells at the same time. Introduction of biochromatic or two-color version to allow simultaneous detection of distinct type of antibody isotypes or cytokines. And lastly, the use of fluorochrome conjugated secondary antibodies to allow direct visualization of spots and eliminating development step. And lastly also the introduction of the automated ELISPOT and FluoroSpot readers for rapid high throughput digital enumeration of spots. So those are the things that have come up in the last 2 decades. And this is just an example of how fluorochrome's FluoroSpot looks like. So you can actually detect both interferon gamma and granenzyme B in the same ELISPOT well. And here is just looking at both of them. So some of these cells are producing both of the cytokines when you look at it right here. So this is a very useful technique to detect cytotoxic CD8 T cells. And this is just references of most of the stuff that I showed you today. The paper that came in Nature in 2013, and this is my paper that I had in January of 2013, my previous lab. And if you have any questions, I'll be happy to answer. Thanks.